So Professor Weinberg is one of the leading scientists that enriched our knowledge about cancer and its initiation and progression. Robert Weinberg is the founding member of the Whitehead Institute for Biomedical Research and Professor of Biology at the MIT. He was awarded several accolades, including the inaugural Breakthrough Prize in Life Sciences, Wolf Prize in Medicine, National Medal of Science, and many other awards. He is also an elected member of the Royal Swedish Academy of Sciences, National Academy of Sciences, and a fellow member in the American Association of Cancer Research Academy and the American Academy of Arts and Sciences. First, I would like to thank you for your cooperation and accepting our invitation, and we will ask a couple of questions about your career and about cancer in general. So the first question...
I look forward to speaking with you.
Thank you very much. So the first question is, in layman terms, what is cancer and how does it occur?
Our bodies are made of a large number of normal cells. The number is 3 times 10 to the 13th or 30,000 billion, which is a large number of cells. Most of these cells have the ability to divide and multiply. And in fact, cell multiplication is so common in the human body that by the time we live to an old age, our cells will have divided 10 to the 16th times. That's 10 million billion times cells will have divided. And each time such a cell divides, there is a danger that one of these cells may begin to divide uncontrollably. And so a faulty cell division will lead to 2, 4, 8, 16, 32 cells under conditions where maybe only 2 cells should have been produced. And therefore, cancer is in one sense runaway, uncontrolled cell division, where ultimately the tumor that is formed may be constituted from large numbers of cells that simply proliferate uncontrollably. Cancer does not come from outside the human body. It is instead a defect that arises within our normal tissues because of this uncontrolled cell proliferation. As these cells continue to divide and multiply, they create tumors of larger and larger sizes in various organs throughout the body. And some of these tumor cells may break away from the initial primary tumor and move to distant sites in the body where they will form other tumors. And these new colonies of tumor cells are sometimes called metastases. To give you a feeling for how large tumor is when it has so and so many cells, a tumor of approximately 1 centimeter diameter has on the order of 1 billion cells in it. So one can realize that even though this is a large number of cells, the total number of cells in the human body, 3 times 10 to the 13th, 30,000 times higher. And even a tumor of 1 centimeter diameter is still hard to detect compared with the total number of cells in the normal body. Important is the mention of the fact that if you look at tumors under the microscope, in many respects they resemble normal tissues, and in that way are difficult to treat sometimes because they are so similar to the normal tissues that exist throughout the body.
Mm-hmm. Okay.
So that's a great answer. So our second question will be regarding the current status of cancer research. Do you think that we are close to vanquishing cancer?
Well, in fact, the lives of cancer patients the lifespan of cancer patients is being increased every year by a bit more. Various kinds of therapies some are moderately successful, some are extremely successful. And what this means is that tumors that might have otherwise killed a patient in 6 or 12 months may in some cases be held under control for a number of years, for example. Still, if one hopes for the victory over cancer over the next coming years, I think that such a victory is an illusion, because the new ways we have of treating cancer are making minor gains here and there, but overall the death rate from cancer has not changed that much in various countries throughout the world, in spite of these new ways of treating the disease. There are exceptions. For example, in the West, in the United States, and in Europe, the death rate from breast cancer has decreased by 30%, and that's a real gain, a real victory. It's not 100%, but it's a clear reduction, a very real one. Still, if one wishes to look forward to a time when the death rate from cancer will be greatly reduced, one must be realistic. I was at a conference in Amsterdam about 4 or 5 years ago, where people asked me, how are we going to reduce the number of people dying from cancer? Their goal was to eliminate cancer from the Dutch population over the next 15 years. And they wanted to hear the answers from me as to how this might happen. And I told them, look, to be realistic, if you want to have very significant reductions, decreases in the death rate from cancer, the only way to achieve that is to prevent the disease from forming in the first place. In the United States, for example, we estimate that as much as 65 or even 70% of cancer is preventable. And if one prevents a case of cancer, then obviously one doesn't have to worry about treating it. In the United States, one imagines that 30 or 35% of cancer deaths come from tobacco use. At the same time, many of the other deaths from cancer come from a variety of other factors that are under our control, including obesity, including the eating of improper foods, diets that are low in vegetables and high in red meat, including inadequate exercise, including too much exposure to the sun, and so forth. So to the extent that we would like to reduce very significantly death from cancer, the answer will come from these changes in lifestyle, which are, by the way, very difficult to impose on the general population. People don't like to change the way they live and the way they eat.
Mm-hmm. Okay, that's a very great answer. So the third question is on a more personal note. What intrigued you to pursue a career in cancer research?
Well, to tell you the truth, I began my career studying the molecular biology of RNA and DNA molecules and studying viruses that cause various kinds of cancer in mice. But I was interested in these viruses not because they caused cancer, but because they had interesting molecules associated with them, RNA and DNA molecules. Mm-hmm. Only accidentally, 40 years ago in 1975, did my laboratory start working on viruses that actually cause cancer, and discovered that the disease of cancer itself was extremely interesting. And so people in my laboratory began to explore in greater detail the oncogenes, the cancer-causing genes that were carried by these viruses. And that led progressively in the late 1970s to our studying cellular genes, not viral genes, that are responsible for triggering cancer in our cells. And so that was the beginning of our study of human oncogenes, cancer-causing genes, and in the mid-1980s, the study of the retinoblastoma gene, which acts to suppress tumor formation and acts in an opposite fashion to oncogenes. So I've been attracted to cancer research because it's very interesting and challenging and thought-provoking. And I enjoy trying to solve some of these problems which prevent us from understanding in all of its details the complex disease of cancer. For me, it is an interesting intellectual game. Other people go into cancer research because they want to save millions of people. I have gone into cancer research because I find it extremely interesting and challenging to see how we can devise strategies to defeat human tumors.
It's always an interesting way to be engaged in a certain topic. So you told us about oncogenes and tumor suppressor genes. So could you give us a short account about the story behind this great discovery?
Well, the oncogenes were discovered in an interesting way. My laboratory was working on studying an oncogene that was carried by a mouse sarcoma virus, a virus that induces connective tissue tumors in the mouse. And we understood roughly the structure of the oncogene, and we were also able to take the DNA molecule containing an oncogene like this mouse sarcoma oncogene, and introduce it into a normal cell, and the normal cell would respond, having received this oncogene that was introduced into it. The normal cell would respond by becoming converted into a cancer cell. And this we understood from the actions of this virus-associated oncogene. And once in the winter of 1978, we had a terrible blizzard here in Boston, and we had 1 meter of snow on the ground, and the entire city was shut down because 1 meter of snow, which you've never seen in Iskandaria or Al Cairo, 1 meter of snow is too much to deal with. And so the city was paralyzed for a week, and one day I was walking across a bridge, crossing the Charles River in the middle of Boston, and I had the idea that maybe we could use the strategies and techniques that we had been using to study viral oncogenes to also detect cellular oncogenes. And so we began to work on that in the beginning of 1978, and by 1979, we had discovered an oncogene that was induced when a normal mouse cell was exposed to a chemical carcinogen. And ultimately, we found that gene was a mutant version, a damaged version of a normal cellular gene. The discovery of the retinoblastoma gene was even more indirect. A new postdoctoral fellow came into my laboratory, and he told me he wanted to isolate the gene for retinoblastoma, which operates in a fashion that is opposite to the actions of the RAS oncogene that we had studied previously. And I smiled at him, and I said to him, in effect, he knew no molecular biology. He had no expertise in how to isolate genes. He even knew relatively little human genetics in terms of understanding where genes are located on chromosomes. And so I didn't try to discourage him, but I didn't encourage him either, because I had no reason to think that he had any chance of finding the retinoblastoma gene. And so on his own, he walked across the same bridge that I've talked about before. He walked into Boston, and he walked into the Massachusetts Eye and Ear Infirmary, where there was a researcher who himself was trying to find the retinoblastoma tumor suppressor gene, as it is called. And they created a collaboration. And ultimately, within several months, the man at the Massachusetts Eye and Ear Infirmary, and actually 2 postdocs in my own laboratory, they, through a stroke of good luck, isolated the retinoblastoma gene. I must say this is really what happened, and I cannot really claim credit for it. It happened in my laboratory, but it was not my insights and my encouragement that led finally to the isolation of this very important gene. Even though everybody credits me with its isolation, I had only indirect involvement in the isolation of this gene. So that's a less than exciting story, but that's the actual true history. Later on, in the early part of the new millennium, in the year 2004, I had been encouraging people in my laboratory to look for metastasis genes, genes that enable carcinoma cells to spread from one site in the body to another. And in 2004, they actually discovered a gene that had exactly those properties. And there I once again played an important role in encouraging them to find such a gene. So the route to discovery is very different each time one makes a discovery. Sometimes the head of a laboratory like myself is deeply involved in the discovery. Other times the head of a laboratory only has an indirect involvement in the discovery of a new gene. Okay, Hoyas?
Yes. That's an interesting story, actually, with some details that were not known for me personally. I don't know about Taha. Okay.
Yeah, that's new to me.
Some of our fans are actually young researchers who want to pursue a career in scientific research. What kind of advice you want to give them?
I'm sorry, you're breaking up. I can't understand you very well. Can you repeat that, please?
Yes. Some of our fans for the I Believe in Science page are young researchers who want to pursue a career in scientific research. What kind of advice you want to give them?
Well, first of all, if you want to pursue a career in scientific research, you need to enjoy doing scientific research. Some people want to become researchers because their parents encourage them to become researchers, and that's not a good reason. One needs really to be stimulated by the research and to enjoy doing the research. That's critically important. The additional thing is one can't just jump into a laboratory and begin to do experiments. It's really important to know what the important questions are and the basic underlying principles. When somebody comes into my laboratory and starts research, they will previously have had some academic courses in cell biology and biochemistry and genetics, so that they can begin to understand what cancer really is and what kinds of experiments are really practical in terms of posing specific questions. That leads me to another point. If you ask how many things are there to learn about cancer, there are more things to learn about cancer than there are stars in the sky. And so the number of experiments one can do is literally infinite. And now one has to focus on the really difficult question, which experiments are important and which are relatively uninteresting, which questions are important to pose, and which questions really have little consequence in terms of leading to new insights into how to detect the disease, how it develops, and how to treat it. There are some people who do all kinds of research who simply like to gather data. They gather information on lots of things, including, for example, measurements of different kinds of cancer cells. And you ask them, why specifically are they doing these experiments? And they have a relatively difficult time explaining why they're doing the work. And this really reveals the critically important role of posing important questions at the beginning of one's experimental career. Which important to answer, and which experiments really are of secondary interest. One should therefore avoid at all costs gathering data, gathering information simply for the sake of accumulating data and information. One needs really to have a clear view of important questions to be asked and to be answered. Of course, having said that, one has to add in addition that if one has figured out important questions that can be asked and answered, one has to also ask which of these experiments that are suggested by these questions can actually be done. Because many experiments can be conceived of, can be planned in principle, but when it comes time to actually attempting to carry them out, the techniques, the technology that we have at present may allow these experiments to be done very easily, or in contrast, they may be very difficult to carry out because we don't have the right experimental tools, the right instruments, the right strategies, and so forth. So there's a complex set of decisions that should motivate students who want to begin cancer research into choosing the problems they should undertake and can undertake successfully. Does that begin to help explain or provide them with some insight?
Yes.
Okay, so engaging in scientific research seems challenging, and many of our followers can't speak English. Our community, I believe in science, is based on helping them to engage in science. So do you have any words for our page and our website community, I believe in science?
I'm sorry, I didn't understand what you said. Is that Mr. Abdul Halik?
Yes.
Yes. So I believe in science.
Do I want to say any words? Science page.
Its main purpose is to,
Yes.
Exactly. Yes.
So, well, let's say we're talking about young students in Egypt. What's important for them is to really get an excellent education in the basic sciences that support cancer research. As I said previously, to get a good education in cell biology and biochemistry and genetics and even molecular biology. That's very important. And as I believe you may also have mentioned, in the English language. English is not necessarily a better language than Arabiya in terms of understanding science, but for various historical reasons, English has spread throughout the world as being the international language of science. And therefore, it's very useful to learn that language, both in the written and spoken form, simply because many of the important scientific articles are written in English and require a reading ability in English, in the same way that many of the scientific conferences that are conducted throughout the world are held in English, again, not because English is necessarily a better language than Chinese or Arabic or German. It simply is the case that English has become a currency that is used by scientists throughout the world. Moreover, one might say that students who believe in science should try, at least for part of their careers, to get some training outside of Egypt so that they can learn different ways of approaching science and return back to Egypt and enrich the group of scientists within Egypt who are studying the disease of human cancer and who are attempting to treat it, to cure it in the Egyptian population.
Mm-hmm.
So that was a great answer, and we would like to thank you for your precious time. And any words, Mustafa?
I just do you want to say anything to the fans who are actually following the page, whether on Facebook or actually reading the articles on the website? Do you want to say anything regarding the website itself or the people who are working on it?
Well, I think it's remarkable that you're establishing this website and that you have a large number of fans who are following this website. This is the beginning of starting a new revolution in science in Egypt. If you can gather a large group of scientists, of young people, who are really truly interested in basic research, scientific research, and who have the understanding that if one discovers new basic fundamental truths about how cancer arises, will sooner or later gain new methods and develop new techniques for treating this very difficult disease. So I wish them and I wish the website community much success.
Thank you very much.
And I thank you for allowing me to speak today. It's my pleasure. Afwan.